Stuff that'll be toward the end of this section, but everything kind of up to that point is like factual anatomy, and we will have pictures to kind of show you guys, give you visual aids, and we'll kind of just dive into it. Um, in order to understand nutrition and feeding mushrooms and mycelium, it's we just have to understand it, right? We have to understand how the mushroom functions, how mycelium grows, what it's doing, how it's doing it, and it really unlocks the foundational understanding of what we're feeding them and why. It's not even the what, because you can find the what anywhere, right? No one came here today to like take the class or hang out with us because of what. I mean, we all know what to feed them. If you Google it, you'll find recipes, but it's really why. So, which came first, the mushroom or the spore? Any theories? All right, so we'll go over this little basic anatomy chart and just kind of walk through this real quick. We're not going to spend a lot of time here, but it's just to give you visual aid for anyone who's new to this kind of information. It's definitely the spore, came in on a comet, uh, or maybe it was here from the beginning. Um, all right, so Basidiomycetes as a group of fungi are like guild mushrooms primarily. Just because they produce basidium does not mean that they're going to have gills. There are polypore mushrooms that are technically Basidiomycetes, and inside of their polypore tubes, they produce basidium that produce spores. So we will start at the mushroom because it's the most visually adaptable thing. So anyone that knows anything about a mushroom can knows what a mushroom looks like, at least a basic figure of it. So on these gills, these gill walls, they produce these things called basidia or basidium for singular, and they produce spores. This is their reproduction mechanism. And in the early stages, when this is developing, it's called a zygote, and they produce these haploid nuclei. Nuclei are just a fancy word for um, nucleus, which contains the genetic and DNA information that it's passing on. Okay, so to not overthink this and overcomplicate this, these are basically cells that contain the genetic information. Okay, so the zygote, when it's forming, it does produce these two nuclei that come into it. These nuclei split and form four haploid nuclei, and then it will produce something called a sterigmata, which are these little spikes, and that'll be produced first on top of the zygote or basidium. And then through the sterigmata, these haploid nuclei will be pushed into and form a spore. And specifically with cubensis and psilocybe mushrooms, they are they produce four haploid nuclei spores. So they produce an equal amount of spores. So these spores will go off into their environments or into our swabs or foil and get grown out. And once the spores are germinate, they will germinate something called monokaryotic mycelium. We'll get into more details here. These two mycelium mate. They produce what's called a Dikaryon, and this will fruit mushrooms. Okay, so that's like a really basic overview. It does cover most of this information, but we're going to kind of like dissect that a little bit more and really, really dive into it. So, and these are actual images of what this looks like on the side of a gill wall. Here is our zygotes. You can see some in the background where they actually are producing their sterigmata and forming early formations of what a spore is. And once they fully develop this, it will mature into what is a basidium with four spores, an equal amount. Now, depending on the mushroom, there's a big change in how many basidiums that are actually mature versus not mature. So when you're working with lab genetics, something that's like bottlenecked, mutated, or whatever, We'll have something called cystadia, which are like sterile structures. So some of these little lumps we'll see in the background will never actually mature into a basidium. That is because they're not able to. So um, that usually just means they're like sterile cystadia. So cystadia is just like a term for a sterile feature, which looks like a basidium quite a bit actually through a microscope. Yes, you can totally swab unmature spores. Absolutely. I mean, there probably happens anytime you swab anything. There's probably going to be some, but like literally in a square inch, there could be a million spores. Especially with something that's like really has a lot of sporulation fitness. They can produce incredible amounts of mature basidium with tons of spores. So if that is something you're worried about, picking up unmature spores that aren't going to be good for you moving forward, I wouldn't worry about it at all. All right. Each spore also contains its own sexual orientation. Here's some morphologies or like phenotypes of cubensis spores. We have three different distinct features. We have albino, yellow, golden, rust, whatever you want to call it. I've scoped every single one. I've never really discovered something that was truly a different color in that spectrum. And then we have purple. So where the color variation comes in is usually based on the density of the spore print or density of spore production, right? So we usually have I have I think 7 rust strains. I like calling them rust strains. It's just always what I called them. But their spores are typically yellow when they're alone. They're very yellow or golden, whatever you want to call it. And as they pile on top of each other, they become more dense. And because they're so small, they allow light through because they're pretty transparent. They're like little pieces of stained glass because they're so small. So the more of them stacked on top of each other eliminate how much light or limit how much light is passing through. So therefore they can look darker. So sometimes it's where we get this like rusty brown color. Even with our purple spores, sometimes they're more toward a brown color versus purple. So they do kind of fluctuate in color, and a lot of that is based on density. Because if you have something that's like a classic mushroom, is very high in spore fitness and drops a lot of spores, if you leave it sit and sporulate onto a piece of foil for a very long time, those spore prints will look black. So just something to be aware of. All right, this is like my favorite slide. I love sharing this with people because it's just awesome. It's so much fun. So this is like a 2500x high definition photo of a spore with its internal anatomy. And we can really look at it from its iconic shape. It looks like a football. It is concave on one side, which is indentated. So it's got like a bowl shape on one side, and it's convex on the other, where it's very rounded. And it is really important to understand that like nothing in nature forms nothing in nature is formed in a way where it's not of use. Nature just doesn't do that. So this is all very much by design. Endearing these essential traits, they really are essential because we can go back 2 slides and we can see. So we have our rounded edge, and then we have our concave edge on the inside where they all face each other. These little bowls all face each other. We can even see it in this little drawn diagram. And that's on purpose because when a mushroom is maturing on the ground and the cap opens, and we all know mushrooms come after rain, right? So they're always growing in high humid environments for the most part. So it's at least what we're talking about today, like psilocybe mushrooms and most standard Basidiomycetes, common guild mushrooms, they usually grow with rain. So some sort of form of water is available, usually higher humidity. The cap opens, it acts as like an umbrella catching water vapor that is rising off the ground. These little tiny water molecules actually will stick to a spore because spores have a small electric charge, and this little tiny droplet of water will fill that concave indent, and it causes a tiny shift in air, and it allows that spore to release off the basidium and fly off into its environment. And this is, we call this the buller drop. So that droplet of water that fills the spore is called a buller drop, which was discovered and observed by a mycologist named Reginald Buller in like early 1900s. It's really cool. There's actually a video. Buller was a total gangster, man. I love that guy. Like he's of all the famous mycologists, he may be the most influential one for me personally. Just dude, he's like 100 years ahead of his time. Is the stuff he was documenting and observing was just unbelievable. And he doesn't get mentioned by anybody that's a professional mycologist. Like they want him to be forgotten about. Like don't look at him. But he there is a video online. If you search Buller drop, you will find a like time lapse camera, like high speed time lapse camera of a Buller drop actually releasing a spore off of Basidium. It's like the most unbelievable piece of footage I've ever seen. So this is how spores are released, typically. Okay, they not all mushrooms like eject their spores. They're not like dropping them. It's actually humidity. So it's all this like perfect function. So when you drop a mushroom cap on a piece of foil, that mushroom cap is 90% water. So there's this level of humidity that's between your foil and the mushroom cap itself, causing this little humid environment, right? A little microenvironment, and those little tiny molecules of water touch the spores and allow the spores to drop. And it just rains spores, you know? So, okay. Most anatomy of agarics and Basidiomycetes spores are extremely similar. For the most part, I have never observed a single Basidiomycetes spore that is not transparent or translucent. The closest thing to a non-transparent spore I've ever observed was actually Paniolus. They're so black and dense that they do look like air, like light doesn't pass through them, but they are still transparent. They're just less transparent. And I've collected different, you know, mycorrhizal fungi, all sorts of crazy Basidiomycetes on spore, and observed all of these things over the course of years, and I've never seen anything that out of the woodwork. So they're all very similar. All right, so well, what we really want to know is how the spore functions, and in order to know that, we have to kind of look inside, right? We have to look at the anatomy of the spore, and in this diagram, we're going to break this down right now. In this big center, we have the nucleus. This is our dense organelle. It is present in all eukaryotic cells. Your eukarya or eukaryotic cells are multicellular organisms, right? So I think it's called perikaryotic cells are singular cells, like viruses and stuff like that. So eukaryotic cells are multicellular. It's the stuff we are in common proximity to. We're constantly surrounded by them. So fungi, plants, all sorts of things. Typically, a single rounded structure bound by a double membrane containing its genetic material. So it's just basically a genetic sac, right? It's the little nucleus in the center. It contains all of our genetic information. And this is eventually what will grow into our mycelium. And two, we have mitochondria. They are peppered through here, but mitochondria are basically little micro machines. They're most commonly found in eukaryotic organisms, but they are for chemical energy, so chemical synthesis, and they do not do their job without the help of other organelles. Mostly, we'll get to those like rhizomes and nutrient vesicles. Three, we have a lipid drop. This is a reservoir of natural lipid oils. This is basically food. So it's used for cellular energy, but it is like a built-in uh, triglycerol synthesis. So it's basically just built-in food, right? So it's a lipid oil, or commonly referred to as a lipid drop. And with the help of the rhizomes in mitochondria and nutrient vesicle, it is actually absorbed in given to the nucleus, which will form mycelium. Rhizomes are really small. They're kind of peppered through here as well. We have a good one over here. Right here, we have a little rhizome. It's real small. These are macromolecule machines found in all living cells, and they're responsible for the biological protein synthesis. So is the this is the little machine, essentially biological machine, that is responsible for linking amino acids in nutrition together, and it's able to process that synthesis, right? And then it works in tandem with mitochondria. And then third comes in our nutrient vesicle, which is V. So we have these right here. Of course, this is placed right next to our nucleus, letting somebody in. All right, so nutrient vesicle is involved in our metabolism. A nutrient vesicle is basically the transportation of the broken down enzymes that feed the necessary organelle. So in this process inside the spore, the nutrient vesicle is delivering the synthesized lipid drop that the rhizome and the mitochondria have synthesized into food, broken it down, attached those amino acids, delivered to the nutrient vesicle, or the nutrient vesicle delivers it to the nucleus, and the nucleus is allowed to grow. Down at the bottom, we have a hilum. This is the little indentation that is left from the spore releasing from the sterigmata, which we saw earlier. And finally, at the very end of the spore, we have an indentation called a germ pore. This germ pore is what will open up and allow our mycelium to exit upon germination. So once all of these pieces work in tandem and we have mycelium forming, the germ pore will open and mycelium will crawl right out of it. All right, now let's talk about the spore wall, because again, yeah, this is hundreds of millions of years of evolution built into these mushrooms, and everything has a purpose. So the spore wall is actually made of, it's pretty complex. So they're these little microfibril cloth, if you will. It's like a fabric, and they're made of five different layers. And we'll just kind of roll through them: the endosporium, episporium, exosporium, ectosporium, and perisporium. And basically, what happens, these are made of chitin and B glucans. That's their composition. The um the composition of them is, let me see here. Sorry guys, I'm just trying to minimize this window. There we go. Boom. Okay. So the composition is important because we're going to see this kind of get repeated, and I just want to kind of put that out there so you guys are paying attention to chitin and B glucans. We're going to talk a lot about that. So the outer layers of the spore are all environmental based. These spore layers act as environmental triggers to start this process, right? So we walked through what all of these organelles do, how they function, what's actually happening inside of the spore, and what triggers that process? Moisture. So when the spores, if this mushroom is growing on a cow patty, like a classic cubensis, it's in the wild, it sporulates, spores get dropped off into some grassy field, and there's cows, and cows start eating this grass, and it takes in the grass into its stomach after it's chewed it up, and it has cubensis spores in its warm, hot belly full of grass fibers and all of this vegetation being broken down. That is the perfect environment for these spores. It's probably 90 degrees, 80 degrees. It's very hot. It's full of the things that they need and require for nutrition. And the cow defecates on the ground, drops a big turd, and are spores able to germinate because it's encased in its manure. So if there's, I'm sure all of you guys have seen cow manure at some point in your life, or horse manure, and so it's pretty large, and the spores are protected from the outside environment. It's warm, it's damp, they're inside of it, really high CO2, not a lot of O2, right? Not a lot of oxygen. So That's the perfect place for this. And as that moisture signals to these spore layers start softening and they start kind of like disappearing, right? So they soften, kind of become gelatinous. And that's what allows the internal process to start. Yeah, I wouldn't doubt that spores can germinate on gills. Like EOL is a big thing. I'm not gonna lie, I'm gonna be totally honest. And I see so many people post pictures of mushrooms that I would never touch. Like they're far beyond. They should have been harvested early. They're kind of like blue gills. The gills have filled with fluids and they're just old and kind of mushy. I often refer to it in short as EOL, which is end of life. So we start the end of life cycle. Once the end of life cycle starts, there's rushing amounts of the cells in the mushroom tissue fill with fluids. This is what causes oxidation. So when your mushroom starts oxidizing, you get these blue nipples or they start softening. Like that is the beginning early signs of EOL, right? So end of life cycle is starting. You have 24, 48 hours and your mushrooms are nothing but mush, right? So they turn into like really soft. The problem with this is that if they are being grown for consumption, is that those mushrooms are degrading, right? So when they start filling with fluids and they start oxidizing psilocybin, um, it's kind of at the end there. To answer your question, yes, we're going to be posting this all over the place. I'm going to try and post it up in as many platforms as possible. Yeah, it could be a vector for stagnation. Too much water, rot. Yeah, you can get rot. I would find usually if your grow is too humid, mushrooms become really lazy, so they don't grow as well. If you're too heavy on hydration, they grow slow. The mushrooms don't usually flush as well. But the trick with high hydration is just airflow, right? So if you can maximize airflow with maximized hydration, you're going to get big mushrooms. But yeah, I do think that spores can germinate on gills. I do think it's possible. We see mycelium form on caps of mushrooms. I've cloned dried mushrooms and all sorts of stuff. They just want to grow. They're so adaptable and so set for survival. Like there really are of like almost perfect design, right? Like I don't really see how you could hurt mushroom in many ways. They actually I think they're so well designed and equipped for survival that if you hurt them, you're actually helping them. It's like I always think of this story of my buddy that lives in Washington State and he goes to the dunes looking for azurescens, and he said that a few years ago he was telling me that the local police were like really bad, like they were like hiding in bushes and shit and trying to jump people, and like oh you're picking mushrooms, you're going to jail, and like he said he really got weird for a little while there because local it was always just known like they were there, they just grow there, it's no big deal, and but he told me he would walk down the beach with a golf club and he got stopped by some police and they were like, "Hey, what are you doing? Are you picking mushrooms?" And he's like, "No." He's like, "I hate these damn things. I'm killing them." And he hit him with a golf club. And the cops were like rooting him on, like, "Yeah, fuck those mushrooms." And he's just sporulating the whole field. I just think it's brilliant. It's just great. That's the wonderful thing about the mushrooms. They're just so resilient. Their survival mode is there, you can't stop them. They can't. They're millions and millions and millions of microscopic spores. It's just hilarious. There's no way to do that. So yeah, they're just so highly adaptable. It's amazing. Monokaryons. Okay. Oh, this is weird. I swear there's a missing image here. Oh, okay. All right, cool. Never mind. Back to it. Spores to germinate. We will talk about that. I promise. We'll get into the germination stuff. We'll do some germination conversation. Spores absolutely can be stubborn to germinate, especially lab stuff. It just happens. All right, monokaryons. Psilocybe cubensis, particularly all psilocybes, actually, have two different kinds of mycelium. Our very first mycelium is called monokaryotic mycelium. And this is single spored mycelium, okay? And then we also have dikaryotic mycelium. Spores, we have, as we've talked about already, they have different orientations, so they need an opposite to mate, right? There's male and female. When hyphas start to emerge from the germ pore on the spore, which we talked about, right? So when that mycelium finally germinates, comes out of the spore, that single spored mycelium is considered a monokaryon, or it is monokaryotic, right? So those both being single spored mycelium. Um, Now, single spored mycelium is not capable of producing fruit bodies. It is very difficult to view without magnification equipment unless you are constantly working with monos. It is just not easy. So if this is something new to you, you've never thought about working with monokaryotic mycelium, there's a lot to learn, a lot of trial, a lot of error. Obviously, if you really love what you're doing, you're going to figure this out. It's not going to be a problem for you. Dikaryotic mycelium is what we're all used to. We're really accustomed to seeing. It is a mated of two compatible monokaryons that have mated and come together to create a dikaryon. Now, all of psilocybes are considered heterophalic, so they do have a plus or minus, male or female, whichever you'd like to call it. I don't think it's so much of it being male and female per se. I just think that it's of different orientation. So I always like to use that as a descriptor. It's like male and female because they are considered technically by technical terms heterophalic, which means they're that the heterosexual, right? So heterophalism, but the technically in a way though it's kind of just our way of describing it, right? As humans, it doesn't mean that they are really female male. Maybe it doesn't work that way, right? It's just about, I think it's a pheromone response personally. I'm sure in the next 10 years we'll have some sort of high-tech equipment that'll be able to detect these kinds of things. Tetrapolar, they're not tetrapolar, they're heterophalic 100%. Tetrapolar is something I've seen get floated around in conversation about psilocybe mushrooms. Um, it's just not the case though for these. It's just not, unfortunately. So, they do,
Bass,
yeah.
Bass, we do have a question. Um, Someone asked, what if spores are stubborn to germinate?
Yeah, we'll hit them in just a few minutes. I want to get through the next
All
two
right,
slides. So and then I'll touch on spore germination because we're gonna do some germinating of spores. We'll do a couple of texts
awesome.
here too. Then we can talk about stubborn texts and ways to go about dealing with that. So breeding pairs, yeah, you can breed a lot of different pairs, and we will get into all sorts of different forming of breeding and how that works naturally. And you guys can use your imagination, put the dots together. This class, I have a really firm stance on not teaching specific breeding techniques because I don't think that people who don't deserve it should know. But if you know what you're doing and you care and you're here, it's here. Just pay attention. If you're, that's been my key for teaching this. That's why this hasn't been, um, God, I love mushrooms so much. I just don't want to see people ruin it. Does that make sense? Like, there's parts of aspects of it that I love so much that I can't just say, hey, this is how you do this step by step, even if it's ultra difficult, because there's someone out there that doesn't care about it like I do, and they come into this for a different reason than I do. Um, so for that reason alone, if you guys can read between the lines, please read between the lines. If you can't, this class is not going to teach you how to breed, period. And I'm not going to teach you how to breed because that's not what this is for. This is for giving you foundational understanding that you can take away and you can apply that to how your approach is. Because then once you have that information, you can build off that, right? So the whole point of the class is just to give you this ultra firm foundation. You can stand on it, you can build on it, and what you build on it is ultimately going to be your path. That's the whole point. So All right, so male female monokaryons mate, they become dikaryotic. They do go through this like um, huge nuclear fusion where the donating of nuclei to a compatible mycelium, right? So if for layman's sake, a fertilizing male would donate a nuclei and migrate it to a receiving female mycelium over what is called a clamp connection. So they would bridge a connection to each other. Then it would grow this little clamp connection where they actually become one. And they start trading cytoplasm, trading organelles. They become literally one. They become fused together. And in that nuclear process is just them breeding. That's their fertilization. So the female becomes technically fertilized, but at the same time, they're actually becoming just one. Um, there are studies, though, that have hypothesized specifically Basidiomycetes, Schizophilum commune. It was a work study I read on that mushroom. If you guys are unfamiliar with Schizophilum commune, it is a little white mushroom that grows in the winter on usually fallen sticks from trees. They're really pretty. They look like little snowflakes. They're beautiful. There's actually a lot of research on this mushroom, oddly enough. But because it's a Basidiomycetes, we can kind of already formulate that a lot of the information from the work study is probably transferable to a lot of other Basidiomycetes, right? And if you watch and observe, you can actually see that in action. So they did hypothesize that the female or receiving mate can be a little selective. And we're going to get into like polybreeding later and way late in the class, but we will talk more about this. I just kind of wanted to point it out because I thought it was a really cool thing. So, like most mammals and other creatures on the planet, fight for breeding rights, it is possible that mycelium and mushrooms are doing the same thing. Okay, monokaryotic mycelium. Here's a great shot. This is what it looks like. There's a spore germinating, and it's growing out this singular mycelium. There is these little indentations are just like cytoplasm in organelles. So there's cells moving through the mycelium. And if you can tell, one of these spores must be getting ready to germinate because it grew directly to them. So the fact that they have anyone that would think they're not capable of sentience or having some sort of signals, right? So whether that's pheromones, something's going on where this is it, right? So they know it knows it's waiting like, yo, what's up, baby? Let's go. They're ready to rock. So monokaryotic mycelium again, single spored mycelium. We're gonna go in. We got our different orientations. We're gonna get mated. Again, monokaryotic mycelium does not produce fruiting bodies. So if you start working with monokaryons and you produce a fruit on your plate, you do not have a monokaryon. All right, clamp connections and nuclei. Dikaryotic mated mycelium of two compatible monokaryons. These mycelium colonies will come together, they'll mate. You know, we talked about heterophalism, the nucleus donation, fertilization. It all happens right here over this clamp connection. So if you are using a microscope in your lab to confirm your mycelium is bred, living, viable dikaryon that will produce fruit bodies, this is how you're going to see it. Primarily, what you'd want to be doing this with is if you are working with monos and you want to verify it's a mono or not. So that's really what we're looking for. Now, if you're not wanting to get that advanced and you want to, let's say you're really just trying to learn the observational side of this, I would urge you to do that. So if you're not trying to breed and you're not trying to do something super advanced, just seriously, if you got a microscope, use it. You know, take time out of your life and sit down for an hour and just put down some mycelium. Keep putting it down on slides until you get a great shot where you can really observe. You can really watch the mycelium and see how it grows, and get an eye for spotting these details. Look for these clamp connections. This is a aerial view above of those same clamp connections. They look like nucleus, right? They look like a nuclei stamped into the mycelium, and they're actually just clamp connections. And we can see them all over the place. So, a really cool thing that I'm going to mention is that, we were talking about survival adaptations of mycelium mushrooms and just how unbelievably resilient they are. So, by design, they are built for catastrophe. The septa is below the clamp connection. So, here's a septa right here. Um, this is your septa right here, and that's literally what I'm about to talk about. So, mushroom mycelium grows in these tiny little segment forms. It's ultra microscopic. We're looking at 2000 to 2500 X. I don't really remember what this was taking it specifically. I'm going with 2000 X. So we're way in there, right? We're way zoomed in. And from one clamp connection to another is just a fragment of a millimeter, right? So it's super small. And within this frame of growth, every single building block needed to regenerate and grow itself is all right here, right? And so for all the people in the class who have actually grown mushrooms, just think about when you break up your grain jar or your grain bag and watching how all these different little inoculation points fuzz up and reconnect and colonize to each other. It's only possible because of this anatomical feature where between one septum and the other septum, and these little septums are doorways. So these septums open up like a shuttered door. So if you are, I don't know, anyone's watched like sci-fi, Star Trek, something like that, the spaceship cliche door that like opens up and closes really fast. A septa is basically that. It is a doorway from one section to another section. So it allows flow of cytoplasm and organelles to this doorway. The doorway opens up and allows it to keep flowing. And the reason it's flowing is because mycelium is doing what we were talking about the spores doing, right? The mycelium has cells that are carrying nutrition different places. All right, Dikaryons, just to finish on the Dikaryons. Dikaryons are mated, visible, fluffy, white, bright mycelium. They don't mix much, and they are capable of producing fruit bodies. And we can see their incompatibleness with each other, where they create these division zones between colonies. This plate's an absolute cluster of a mess, but it's a really great observational tool to use. So once you know that there's a mated Dikaryon, usually they kind of look like this little plume where there's the tiny white dot in the center. This is a brand new Dikaryon. Now, there are a few in there that are a little harder to see. Honestly, they really don't really show up very well here, but there are some, and that's kind of what most Monokaryons look like. This outer growth that's kind of gray, sometimes translucent. I heard someone referring to it as like scout mycelium at one point, but it's not. It's either enzymatic issue where it's having trouble absorbing nutrition and growing, or it's a monokaryon. So they're usually very light, different colored, and they're very, very, very slow at growing monokaryons. Very slow. 20, 30 days to colonize a full plate. Um, I have a lot of monos, and I don't have to touch them. They just sit there stagnant. But to answer your question on the last photos, those were 2000X. So if you guys are in the market for buying a microscope, get something that goes in at least 2,500 times with an oil objective. All right, what's going on in our mycelium? Let's talk about this. So just like our spore, we have the same group of organelle structure, kind of. For the most part, the same organelles are still present. So we have the same, we got a nucleus that's floating around, mitochondria, nutrient vesicles, all these things. So most of the materials that are exported most of the materials that mycelium come into contact with are exported by something called the endoplasmic reticulum, which is the tip of the mushroom mycelium, also referred to as a Spitzenkorper. And at this tip of the mycelium, we have something called Golgi organelles. These vesicles Basically, what they do is they're delivered from the apex of the hyphae, and they're delivering nutrition throughout the rest of the
Uh,
through the rest of the body. So, um, so this process is called, we're going to go through these two different processes of it. The membrane of the exterior cell wall is made of B glucans. See, there it is coming up again. We got B glucans. That's its molecular composition. It has 1 in 3 B glucans. It's also made of chitin. Those are the two big heavy hitters there that are something you might want to remember. And they are the composition which is necessary for making those linkages to their preferred foods. So, kind of like we talked about our rhizome earlier, being able to have like nutritional synthesis where it was able to link amino acids. These are just cells, guys, so little molecules, if you will. So they're smaller than cells. And imagine there's a chain that kind of links these little molecules together, and that is how they break down and bring in their food, literally at the molecular level. So these two compositions, chitin B glucans, especially 1 and B3 glucans, are the most common type of polysaccharide found in nature. They're in all sorts of stuff. It's a wide variation. We'll kind of go into that a little more in depth in just like a slide or two. So, so these cell walls of mycelium, though, they're super adaptable. They can grow in three-dimensional space. They grow in 3 dimensions, up, down, left, right, all at the same time. If you guys, are working with grains and grain bags, you probably have seen your three-dimensional growth I'm talking about. You drop a plate in the middle of a grain bag and watch it grow. It's going to grow a softball. It's not going to grow a flat line like we see on petri dishes. So it will grow three-dimensionally, flank out like a ball, which maximizes its digestive productivity and colonization productivity. So during this process of eating, we'll just say, for layman terms, um, there's two processes in their digestion and eating. So first process is called exocytosis. This is where our cytoplasm, and cytoplasm, guys, just for an analogy of sorts, is like mushrooms, mycelium's blood. It's like a fluid that runs through the mycelium, moving cells in and out and throughout the mycelial body. And that cytoplasm delegates nutrient vesicle clusters to a region where it knows there's food to discharge enzymes. These enzymes come out of the cell wall and start breaking down the nutrients that it wants to absorb. So, like oils, proteins, minerals, those kinds of things, it's breaking those down, creating those little chains across those molecules. And then in the next step, endocytosis is the process in which those enzymes produced in the first step have prepared the nutrition for absorption. So these vesicle clusters that are delivered to the nutrition in the spites incorpor, these vesicles are responsible for migrating that distribution of resources throughout the body. So that outer growth tip you see in your grain, you see at the edge of your plates, you see in a ball of mycelium in a liquid culture, that tip of the mycelium is doing all the thinking, all the work, all the signaling. And it's also taking in that food and delivering it to other places in the mycelial body that need the food, right? So maybe some cells that are still active in the center of the mycelium that are far away from where all the action's happening, right? Far away from where all the food is. It's delivering different nutrients there that it needs to stay alive and healthy. All right. So that is what's going on in your mycelium. All right. So we got a pretty cool picture here of some mycelium way in like 2500x. And we can see these little irregular kind of shapes and stuff is because the mycelium is so small, we can actually, with a microscope, kind of see parts partially through it, right? So this is actually, these little indentations are cytoplasm. Like I said, is basically like mycelial's blood, right? It's a fluid that moves cells throughout the body. All right, so this is a really cool clip a friend of mine has let me use for my class. Is a buddy of mine that recorded this, and it's cytoplasmic flow. So this is obviously sped up, but this was taken with a microscope and recorded, and this is mycelial hyphae. And we can see here that this is just the flow of my cytoplasm kind of coming through. That big thing that just cleared out everything, that's called a volcule. Volcules are basically empty space. And for anyone that's like, this is BS, no way. Look at these cells going backwards. Look at that. The cells going against the flow, bringing things to other areas of the mycelium. So they are super active. If you have a good microscope and you have a lot of patience, you can watch cells move. I have one of my microscopes, the one that I said earlier I was kind of frustrated with, is very capable of that. And I have sat and watched patiently, and I've seen these little indentations of cells kind of bunched up, like little round balled cells, and then just sat there for like 20, 30 minutes and watch them inch forward. It's really cool. It's actually kind of mind blowing once you see it. Like seeing it firsthand in person through your own lens is just blow your mind. It's so cool. Um, all right. So, what are 1 and 3 beta glucans? So, B glucans, B stands for beta. These are polysaccharide. Again, it's a common polysaccharide. I'm just going to keep repeating these terms, guys, because it's just stuff that I want to stick. I want you guys to take away and be like, I was learning about polysaccharides. So 1 and 3 B glucans are linear, unbranched chains in kalos. Kalos is a type of 1 and 3 B glucan or a polysaccharide, and the kalos is found in pollen tube walls, grasses, grains, curdlan, paramylin, and pockemin. Pockemin is Basidiomycetes, and I believe paramylin, if I'm correct, I think paramylin is technically crustaceans fall in that. So they're also made of like chitin and 1 and 3 or 1 and 3 beta glucans, or at least partially. There's a really fascinating thing. Polysaccharides, guys, and beta glucans, and the whole subject of polysaccharides is massive, and it's a kind of a hard read. So anyone who's pretty advanced that might be here today, if you really want like something to really challenge yourself with, if you haven't explored, get into some polysaccharide reading and read about them. It is pretty intense stuff. It's pretty cool though. It's a lot of fun if you can wrap your mind around it and you understand kind of what you're looking at and reading. It's a great read. I actually have a great book on them too. Um, I'll see if I can share it in a minute. The specific fungal 1 and 3B glucans, which we mentioned here, are Pockemin. 1 and 3B, they're linear branch on branch molecules in which B glucosal chains, right? These are joined together through these linkages. We've talked about these chained links. These molecules occur in other polysaccharides and proteins. 1 and 3, 1 and 4 beta glucans in cereals, grasses, and embryophytes, which are bugs. So bugs, we always know bugs are synonymous with fungi. We always have, shelled beetles and flies and stuff that are probably used by the mushrooms for spore dispersal, which is pretty cool. Plus many other sequences and linear molecules, the fungi are equipped with making these links in order to synthesize the nutrition, right? So if their compositions weren't aligned like this, they wouldn't be able to create these amino acid and polysaccharide links to break down, absorb, and eat. Um, so what basically all this means is that we need to create a mycelial microbiome because your mycelium is like a living stomach. Now that we know for a fact our mycelium is sentient, it has designation over its consumable nutrition, right? Those nutrient vesicles are delivering food throughout its body. We need to create a biome of nutrition, and it is important to develop a consistent molecule breakdown of nutrition. We know that the mycelium itself is comprised of monopolysaccharides made of 1 and B3, 1 in 3 B glucans and chitin. The B glucans are typical in nature and often found comprised many carb-based plants, right? So insects, debris, this is what we find our mycelium eating in the wild. So in essence, your mycelium is sugar, and it has an affinity in digestion of 1 and 3 B glucans, sugar. So they're carbs, right? What what type of enzymes is it using or releasing? I honestly, to be honest, I don't know off the top of my head. I'd have to look at some of my writing. Um, yeah, do we have like specific enzymes? So so basically, what we are talking about is just creating a nutritional biome of what we're feeding our mycelium. That's correct in within this constraints, right? So it would be something where you want to have it's something that you don't the whole point of this is being like not you're not feeding mycelium something that's completely synthetic, not feeding it things that you don't need to. If your goal is to grow, raise, and isolate healthy mycelium and healthy mushrooms. Now, obviously, if you're working on some sort of science project or trying to find mushrooms that are capable of doing other functions outside of its normal wheelhouse, that's totally different. And that's like setting standards for other research. That's something totally different than what we're talking about. But um, we know that the vesicle clusters are responsible for distribution of nutrition. They develop these enzymes, this enzymatic process, which it's released. They break down the debris, right? So those enzymes are specific to what they're coming in contact with. So cubensis, obviously reliant on cattle, horse, elephants, other bovine in the wild. These animals do not change their diets instantly. So, but you can in the lab, right? You can take a culture that's been raised on malt extract for 10 generations and put it on super low grade agar that's like Gracilaria food grade agar, and then throw it on potato dextrose. And you might have really bad growth. And the reason for your bad growth is that it's a living stomach and it doesn't have the proper enzymes to break down its food, right? So creating these biomes or microbiomes within your genetic is really important. And it's also an essential way to create a cultivar that's capable of fruiting well. So if you're going to work a heavily isolated culture, you're going to work with different ingredients. It is really important that you guys create some sort of biome plan, especially if you have intentions of your genetic being um, capable on other substrates, you know? And when I say substrates, I don't mean substrate specifically. I mean medias, grains, substrates, all the different foods they're going to see. You know, what you don't want to do is bottleneck your genetic into eating one way, because those enzymes have to develop. And here's a great analogy for it, just for you guys to understand fully, make sure you wrap your mind around it. If you are a vegan and you eat nothing but plants, if that's like you're totally plant-based vegan and you've been that way for a few years and someone gives you a steak and you eat it, you're not going to have a good day because the enzymes in the microbiome within your stomach does not have the right enzymatic process to break down that meat, right? It might struggle. You might feel sick. And I've read stuff about people who are like vegan or vegetarian struggling with heavy meat, right? So because they have to take it easy. My brother is actually a vegetarian and started eating meat again, and that was one of his processes where he was introducing it in small amounts, right? Just that upset his digestive system. Even though he was benefiting from it, he said he felt good. It was just the step by step, slowly, right? So And you wouldn't do that from someone the opposite. If you're a carnivore and you're like, man, you eat 2 pounds of meat a day, like you can't go to a strict plant diet. You're probably going to be distended. You're not going to be digesting your food. You're going to get sick. So just think of it like that. It's the same thing. You're stressing your mycelium for no reason. So whether this is strictly just a science experiment for you, or if you're just trying to take care of a good culture, I really think that creating a culture that is adaptable to lots of different ingredients and something that's very complex, has a lot of complex enzymes within its capabilities, is something that I would absolutely suggest to anybody. It takes a lot of work, right? It's a ton of work, actually. It's a lot of work. Like I create um, all the grain I use for my genetics, I use custom made bird seeds, right? And so I make Milo, oats, rye. I have some winter wheat berries sometimes, white wheat, sometimes millet as a filler. So there's a lot of different ingredients you can introduce that are something that you can add a complexity to your culture. So that way, let's say you gift it to your friend in Australia and they don't have the same kind of grains that I have in America available. And they're kind of like we have to use different substrates, right? It would be better to train your mycelium on different things and introduce them all at the same time. So that's where we come down to agar, all of its additives. We're going to start with our base, which is our gel. You know, if you're using media, agar is a heterogeneous polysaccharide from seaweed, right? Um, it is comprised of two different polymers. We have agarose and agaropectin. These polymers are separated for certain applications, but agarose, most of you would be aware, or at least have an idea that agarose is actually clear. It's a clear gel and it's used in PCR work, so genomics, and they use it in their electrophoresis, and this is where we get our barcodes in for doing genome barcoding. And so you don't have to use that for agar though, if you're doing agar work. It's really expensive. It's kind of a waste. So just leave that for electrophoresis. But agar is has two polymers. I definitely would say I would use it in its whole self. It's also a lot more affordable to use. Um, Not all gums, so there are other options, but not all gums will solidify like agar and turn into a solid gel. Uh, but they do offer abilities to change the viscosity or thickness of your fluids, right? So if you're pouring agar and you have a good recipe, your mycelium loves it, but it's just so thin. It's really watery, even if you pour it at low temperatures. It's just watery and it wants to spill and fly everywhere, and you just don't know what to do. You can use a gum. You can use like guar gum, xanthan gum. They are synthetics, but they're very powerful. So you use a fractions of a gram, quarter of a gram, and it would thicken up your agar right away. It would never turn it into a solid. So if you wanted to use it as a replacement for agar, that won't work. But gelum gum will. Gelum gum is a derivative of a yeast, if I'm correct. Might be wrong on that. Don't quote me. I'd have to double check. But it is synthetic. It is made in a lab. It probably a good replacement for agar. Agar, long term, for really huge lab industry, it is a great alternative instead of eating up the seaweeds in our oceans. There are a few different seaweeds, and there are seaweed farms, and there are countries that are their primary exports is agar. So I did talk with somebody who is pretty avid about agar being not good for the environment, but there are really sustainable agar farming practices going on in certain areas of the world. Like Indonesia has agar farms where they're growing agar off of like big ship rope, and it's out in these shallow waters, and they're cultivating it literally for the agar industry. It's used in cosmetics, science, all sorts of different applications in food. So yeah, there's that. Additives. So let's talk about just the nutritional additives. You guys can get super creative with this. It would be something I would highly suggest if you're gonna really play with different agar recipes and just kind of toy with it. Sorry, I keep having this window keeps closing my view here. Okay. All right, okay. Yeast extract, talked about that earlier, kind of on the same scale with peptones. Yeast extract is about 50% protein, 20% glutathione, 6% nucleic acid, and it has at least 18 kinds of amino acids. Potato dextrose. Potato is good. You can also get pure dextrose. I don't think it's necessary. I think it's kind of expensive. You could just use potatoes or make your own potato dextrose. It works great. It's super cheap. It's a great malt alternative. If you're growing gourmet and actives, I would say just stick with a PDA because actives do well on it too. But again, we're talking about creating these like complex microbiomes and stuff you can do. So you can use a little potato and use a little bit of malt, kind of, create that nice complex agar, which your mycelium is going to thank you for. It's going to be super aggressive, grow great. Soy peptones or peptones in general, they are water soluble proteins, protein hydrolysates. They are great. Tons of peptides, amino acids. You just have to watch out for inorganic salts. But they do have lipid oils, vitamins, amino acids, sugars. So peptones are great. Just check your labels, make sure you're not getting something with inorganic salts. Water-based additives. I used to do this a long time ago. I haven't done this in a long time. Honestly, it's just because of the extra time it's required. But if you have the aptitude to do it, grain agar or water agar, a lot of people refer to it. It's just like grain water waste. And you don't really have to use a lot of it. You could use like 100 milliliters of grain water and add in another 900 milliliters, and that's all the nutrition you need to have a successful grown on a agar plate. It's really good for cleaning up dirty cultures. And you can also use, obviously, with coprophilic mushrooms, you can create like broths. So these complex broths, you can make them concentrated, clean them, strain them, boil them down, rehydrate them. And so you get this like really clean, concentrated broth that you can add as an additive to your agar media. I definitely suggest doing that, especially if you're trying to make something, different. You can use 50% grain water. It just depends on how dense it is. Like if you're cooking Milo, Milo is so heavy carb load. Milo usually goes bad very quick. Out of all the grains that everyone's using, Milo is the highest in carb sugars. So it's very sweet, right? It's overload of carb. That's why the mycelium colonizes it so fast because there's so much on-hand sugar. The only problem is that it's sometimes too much. Um, I have found that growing exclusively on Milo is just a great way to ruin genetics. It's nice, it looks good, but long term, I'm talking years. Not this isn't something you're gonna see right away, but like over the course of a long period of time, you're gonna bottleneck faster. You're gonna have issues, health issues with your mycelium. I think the I think the and this is totally anecdotal, but I think that the available nutrition on hand is too high. So, if your growth is fluffy on your water agar that you're using it for, if your growth is fluffy and not ropey, it's too much. So I would dial it back. And if you get too ropey, where those ropes are super separated, yeah, very fluffy, it's too much. Cut it down to like 25%. And then if it gets nice, but it's too ropey, and I don't know if you guys know what a too ropey plate looks like, but this is there. This is what that is. This bottom right picture. These ropes are so thick, it can't even like link together. It's just throwing these like big, huge single hyphase. It's really aesthetically pleasing, but this is not okay. Like this is not good in my opinion. Is it a badass photogenic thing? Yes, of course. It looks incredible, but this culture is like starving. It's starving so bad that it hasn't even finished colonizing the plate and it's absorbing the food coloring. I mean, look at all of these, right? So there's not nearly enough on-hand nutrition, so it's resorting to food coloring as a type of food. So if you want to get something like this in between, this is healthy. It's not eating away the food coloring. The hyphae are spaced, but they're not completely, it's not fluffy, it's ropey. The mycelium's nice and packed together. This is really healthy. It's like dialed in. It's not too fat, it's not too thin. It's probably just a very somewhere in the middle, very balanced. Yeah, yeah, this is so that species of mushroom um has this kind of trait. This is a unique thing to Psilocybe natalensis. This plate here on the left is golden halo, which is a cubensis. Um, so there are a few genetics I'm aware of that have this like mycelial morphology, where the mycelium is almost too ropey. It actually inhibits sometimes their growth. So getting that like somehow under wraps can be a real challenge. I had to work with that golden halo in my peanuts quite a bit to kind of bring in some tomentos a little, just to get them to function and cultivate right, so they fruit better. Because when you have these ropes that grow on top of everything, like this is Golden Halo. I mean, look at this fan at the end. It's like completely elevated and lifted off the plate. And the problem is that's happening on the top of the substrate. So what you end up with is like a half inch of rhizomorphic hyphae on the whole top of your substrate, and it develops primordia at the surface of the sub. So technically the hyphae are interfering with the primordia production, so it doesn't fruit as well. So, and then that's where casing layers, I think, come in and they're very useful. But you don't want to case with something nutritional with an aggressive mycelium like that. You want to case with like pure vermiculite, because it can't really colonize it. It's just a humidity layer. Yeah, definitely. Well, if they're too ropey, if it's super roped out and it's just like looks like it's skipping over the plate, yeah, I would up the nutrition at hair. Um, Bubble wrap, hell yeah, yeah, exactly. I remember people putting a bubble wrap in Orbeez. I seen someone do Orbeez, all sorts of stuff. Oh, the bubble wrap, damn, that is a blast from the past, man. It's like some old school 2018 stuff. Oh, that's good. All right, inoculations. We're gonna dive into some inoculations. Yeah, no, it can't colonize it, of course. Yeah, if you have something that's like roped out, you can't fruit it, bubble wrap it. Like I wouldn't even say that's a bad idea. Where there's a will, there's a way, right? So core is full of kalos. So core is technically really doesn't have a lot of nutrition, right? It just holds water, and actually, it doesn't really hold that much water. Core is meant technically for drainage in plants, right? That's where vermiculite comes in. That's where like manure comes in and other things. Um, Core works great in bags because the bags recycle all the water, so it's always got water. But core is at the end of the day technically kalos. It is plant-based fiber, so yes, the mycelium will colonize it, but it's super low nutrition. If I'm growing in pure cocoa, I'm growing with a high nutrition grain before I completely emaciate my mycelium. Does that make sense? So you pump it up, give it a ton of food, drop it into, a tub or a bag with pure cocoa, and it should do okay because it's had so much readily available food. Now, if you did it the opposite of that, and you're doing something that's high nutrition sub, you want to be low nutrition grain to bring in that balance. Or you're doing somewhere in the middle with everything, right? All right, inoculations. We're going to do some spore inox. They're just little techs, guys, something simple. This is not like ultra insane science stuff. We're just going to do some basic tried and true methods. That way you guys can have some success germinating spores. Early or late casing. I don't case any cubensis. I've actually don't case many psychoactive species at all. I have found that Subtropicalis doesn't need a casing. I have cased Zapotecorums, ATL7 in my wild Tampanensis. I've had success both ways. Um, I've initially cased, then after cycling them 2 generations, I didn't need to case anymore. They were like super adapted to my environment and grew great. In fact, once they adapted and there was no casing, they actually grew better. So I think casing layers are more of like an observational cue when you need it. So if it's something you observe, there's an issue, you're not getting fruits, you're not calling, it's not going into that fruiting stage, air, hydration, everything's right, but for whatever reason it won't fruit. And if you're a few days late, 3 or 4 days, you know that the more time that goes on, that cake's gonna dry out. And the more time that goes on, you run the risk of contamination. So the best thing to do is probably case. That's my 2 cents on casing. I don't think it's necessary and it shouldn't be. All right, inoculation loop. I think I have a missing photo here. Anyways, uh, inoculation loops are really commonly used for like spores. You can scrape spores off. They're really old school. You don't really need them. You guys can use your scalpel blade and scrape spores off from a spore print. It just depends on how sterile you have to be. If you are working in a, if you don't have a dedicated space, yes, inoculation loops can be super useful because you can keep your spore prints kind of like tidy and closed up, and then just grab a little bit of spores on the tip of it, and then, zigzag them around a plate. You can also use it to spread fluids around if you're testing like spore syringes or whatever your vehicle of spore is, outside of like obviously a swab. And so sterilize your loop. I would definitely suggest if you are using these, avoid the throwaways. Just get a metal one. It's reusable. It's easy. You can heat sterilize it. Gather your spores. Either gather your spores on an inoculation loop or drop a droplet of your syringe. You can zigzag them, spread them around. Seal, let germinate, and label your plate. It's kind of a straightforward process. I really, 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 really like spore swabs. I like swabbing prints. I like swabbing gills. I like storing with swabs. Swabs are great. There's a lot of different uses that you can germinate them with too. So not just dropping spore swabs on plates, but I'll cover a different tech. It'll be verbal because I don't have it in the slides. But here, let's watch this video. Spore swab visual aid process. So I don't know if you guys have noticed, but I'm using forceps, and the forceps are heated up in my induction sterilizer that I got from Thermohelix. Those forceps, though, are hot, and I cool them down in the agar, which leaves a little bit of like loose liquid agar, and then that cotton tip sticks to it. So it's just something I really wanted to point out because when the cotton tip's not stuck to it, when the cotton tip's not stuck to the plate, it just rolls around. All right. So now that we got to see some spore swabs in action, Yes, that's a great question. Thank you for asking this question. Yes, so the part of the reason why we're developing enzymes, right? So the question was, if you're not in the chat, the question is, when you talk about mushrooms adapting to media, is that with harvesting spores and breeding again with the next generation? So we pass on genetic markers to our children, right? And that's what spores are doing. It's genetic memory. It's written into its code. So if you grow on certain substrates or grow on all of the substrates, and you have a really adept mycelial colony, and it's seen everything. And let's say you took clones, you weren't going right back to spore, right back to spore. You actually grew a clone out. Clones are usually a little more consistent. They're not as fast. Um, so if you can sacrifice a little bit of time to grow out clones, what you're going to do is have a older culture that performs really well, a little more consistent. It gives you more time to work those ingredients into its genetic memory, right? And then when it fruits, and let's say it's still fruiting good and you don't want it to start going south. Maybe there was something, I don't know. You ran 10 bags or 10 tubs of it, and one or two of them for the first time ever didn't do well. And you're like, oh man, maybe this thing's getting a little tired. Well, take the best one that you have, harvest those spores, because those spores are going to have all of that genetic information already written into them. So those spores are going to perform better that next generation on those substrates that you're using. And so basically what you're doing is you're developing a culture that is domesticated, right? To growing in lab settings on lab medias, artificial grain substrates and cocoa core substrates. They're not going to see cocoa core in the wild. They're copper fills, like it's kind of ridiculous at the end of the day to think about. But now they are. They're adept to growing on them, and you're teaching them how to. We know it works. Other people do it. So that's, yes, a local storage. How do you think your body fights a cold when you're a baby and you've never had a cold? Right? You fight the cold because it's written into your DNA because your grandmother 1,000 generations ago had that same kind of cold. And that's why you can heal from it. It's written into your DNA. So we're doing the same thing with mushroom genetics. Genetics are so fascinating. It's super cool. All right. So curious if you have any thoughts. Swabbed gills of those fresh fruits, potentially offering a clone and picking up gill. Yeah, so that whole end of that video, that's actually what I was doing. One of them was smashed versus like lightly swabbed. And I was trying to see if I was going to get clones or if I was going to get spore growth. Yeah, 100%. If you're just smashing into tissue, you're going to pick up tissue fragments. There's you drop it down, you're going to get a spore growth like this, where you're going to get these segments. You probably are going to find one of these segments is not spore growth. It's probably a clone. It doesn't mean every time you do that and you're doing spore swab work, that's going to happen. But it does give an issue. Does Cabo need to be swabbed? No. Cabo does not need to be swabbed. It's a wild landrace. Drops purple spores. It's just a very classic landrace genetic. Um, the thing is, though, is at the end of the video, you could see I was swabbing spore prints. I really think that spore swabs are super controlled growth. So that's why this photo is here. This photo is literally because of the intuitive questions you guys are asking. Is it picking up fragments of tissue? No, because that is not from tissue. That's not even from a gill. It's from a spore print. So I did drop these here specifically for that purpose to highlight spore growth from swabs, use of swabs, and how that works. The Cabo on the left, I believe that is. I have the chat in front of it. Yeah, so we have Cabo on the left, Cabo on the right. The Cabo on the left is a really fascinating one because it's so uniformly grown. We don't have segments. So we're going to get into cutting plates and stuff, right? We're going to talk about cycling our genetic, cutting them, isolation, what we're doing, that process, at least the broad basics of it. But so Cabo on the left here is super uniform. We don't have segments. Cabo on the right, we have these segments, right? We have these sections or sectors, whatever you'd like to call them is fine. But these multiple sectors technically would be, if they have divide zones between them, which these do. We have a sector area over here, a sector coming out here, a sector here, a dead area, sector, sector. So there's a lot going on this one plate, right? And these all could be potential different Dikaryons, right? So we know two spores mate, create a Dikaryon. So if I'm selecting from what I know is verified as new spore growth, this is what I'm going to take as my verified fastest, most alpha Dikaryon. Now, if I'm taking an alpha Dikaryon, it will likely be like its parent, right? So it's strong genetics, something that's strong, most like its parent. It's going to be the alpha genetic. You're never going to find an albino in an alpha genetic. You're going to find an albino somewhere in the middle of this mess. Those are recessive genes. All right, so yeah, this is uniform, but because it is spore growth, there could absolutely be other Dikaryons. And here's where I'll point out a few. Okay, it's really hard to see, but these are the little intricate things you guys are gonna look for. If you're working genetics a lot, you don't have to overcomplicate it for yourself. So you don't have to do this. I am going to point it out on this plate. There is a divide line. It's right across the center. So there's a divide line right here. And there's a little bit of a divide line here. But it is really pretty uniform. There's also, I would challenge that there's a little bit of a morphology change. We have a little bit of a different morphology in the uniformity of the hyphae growth here versus down here. It's a little different. It's really subtle, but it's not something, it is something years ago I would have super paid attention to. It'd have been like, oh man, I see all these things, these little intricate changes. And I would have like over OCD'd and zoomed in on, right? And these days, who cares? I'll just cut it like 4 or 5 different ways and see who wins, right? You know, like I'm not going to spend my time over analyzing because it's all healthy. It all looks good to me. So right here, I'd be taking a couple big chunks right here. If this looks questionable because it's so uniform, you're like, okay, how could that have happened? There's no way it's a tissue clone. I swabbed a print. Maybe I'll give one of these sectors a chance too. All right, let me see here. All right, let's move on. I was just reading some of the stuff in the chat. Well, nomenclature, yeah, there's definitely a governing body of classifications. That's, yeah, I can put some links in. You guys could read about it. So the question was, just put it on here. Yeah, you touch on phylogenerations in the comments again? It seems possibly to be lab specific, or is there some governing body of classifications? I'm just curious because I've heard a lot of different ways to classify. That's the problem, is that there's a lot of non-professional biologists talking about classifications of biology. Um, Mushroom communities are full of hyperly intelligent people, but we're all, for the most part, self-taught. And this is where biological like structures like nomenclature comes in, or nomenclature, I'm sorry, is essential, right? So you go to these governing bodies that have this stuff set in stone and apply it to mushroom genetics. I will fill in a thing in here. I will find a link. I actually have a link. I wrote an entire like 3 or 4 page blog about it. So nomenclature and how it works and all that. This is a great question though. Thank you for putting it in here. Isolation of multi-spore Dikaryons. So again, we got a plate here of a swab of a genetic that doesn't need to be swabbed. I got really into this for a little bit, and the reason why this plate is such a cluster of a mess is because I swabbed too many spores, and I snapped it off with my forceps in front of my flow hood. And when I snapped it, all the spores plumed right off the tip of the cotton. And so the plate's covered in all these different inoculation points. It's really cool. It's just like sharing this weird little detail. It's just strange, it's it's a unique thing. Okay, So once our spores, once our inoculation shows dikaryotic growth, it is important to have your goals set in place, and we're all talking about interesting things. So everybody's there. Like I feel like everyone who's being active in the chat is there. We're right on point. You're all asking the right questions. We're all bringing up the right topics. Like we're there. So you want to have goals set in place. Like what are you doing with this culture? So when Dikaryons are mated, they're observed. I've observed them consuming other Dikaryons and Monokaryons because they don't move fast enough, right? So, and because of their molecular composition, it is plausible maybe those Dikaryons are eating them, right? They're eating spores and eating Monokaryons that haven't developed fast enough. Get out of my way. This is my area, right? So we can be confident that it is an instant nutritional source for mycelium itself, right? So it's built-in mycelium, built-in food. This is it, right? So being made of the exact same composition required for partial nutrition, these spores, single-spored mycelium, would offer up a great source of fertilizer. So the spores offer a look at a wide range of spectrum genetic pool, right? So we have all these different inoculation points. This is a wide range of genetics of cubensis that are available, and it can vary in results, these are, right, strains, right? So, and they can all vary in results. So we're going to select what we're looking for, right? And on the other hand, rare strains may be left to be overcolonized by competing Dikaryons. It gives you a chance to clone or take spores or take an opportunity where it presents itself here. So what are your goals? Are you going to isolate a advanced, enhanced version of the genetic that you have, right? And if you are, we are going to take alphas, right? That's the point. We're going to take the fastest, cleanest, most aggressive growth. That's how you improve the genetic you're working on. That's how you improve or enhance a genetic that you want to grow, right? So this is AA plus or leucistic A plus, right? By Mr. G. And if I wanted to grow this and wanted big bangers and, I wanted it to be morphologically correct, I am going to take these highlighted spots. Super clean growth. They're more aggressive. They're very thick and white, like healthy mycelium. Over here, this is a reason why not to. We have this clear hyphae. This is an enzymatic problem, right? So it's either a weak strain or enzyme issue. So they're Dikaryons, they're bred, they're trying to grow, they're trying to establish themselves. They're having a hard time colonizing the media for whatever reason. So this is why this is this way, right? So we're going to take something that's healthy, it's colonizing, it's eating correctly, it's producing enzymes. We're going to take something that's super fast, super healthy right here, that's here, right? It's present. That's how we develop an enhanced version or at least a morphologically correct version. Now, if you want to hunt something, you want to hunt something different, something recessive gene or something like that. You don't want to get rid of all this genetics. Like that would be defeat the whole purpose, right? So if it were me, I'm going to grow this A plus plate, which by the way is like 150 milliliter plate. It's massive. I would cut this into 4 sections and colonize different bags. Or I would cut it into like 8 or 10. It just depends on how much work you're willing to do. But there could very well, in one of these weird little weak Dikaryons, be a true albino, or it could be a mutation, or it could be something else, right? So there is a possibility of finding something that's never been seen before within your genetics that you're working. And the really interesting thing is when you find something that's a new morphology or phenotype, and I have personally experienced this where I was working a genetic and a friend of mine was working the same genetic, and at the same time we both found albinos, right? We both were working on leucistic Burma, and we both found albino Burma literally the same week. It's just the most weird thing. And the genetics came from the same place, right? They were both from my original leucistic Burma. So it's really cool that that seeing that kind of happen, where the genetic is naturally moving one direction. And sometimes for that specifically, we're talking about like albinoism. That's that's most likely because we're growing in LED environments, no sun, lack of melanin is needed, etc. So there is some of that, and then of course total quantum non-locality would definitely play a part in that. I think that's just um, too much of something to be called a coincidence. All right. So what is isolation? So isolating, again, in the most broad, basic term, is removing, to isolate. To We're going to remove it and single it out. We're going to isolate mycelium, take a transfer, and create cell divisions of this mycelium to continue our sex selection and force it to expand. And by doing so, we'll be looking for a number of observational cues to guide us through the process. First, we want to wait and let the Dikaryons express themselves the best we can to have a better understanding of what type of speed, vigor that we can expect from it once it's transferred. Some spore plates will express faster moving rhizomorphic mycelium, although in my experience, some tomentose types of mycelium have outperformed rhizomorphic in numerous occasions. This is something that is just, it's totally normal. I've seen it many times. Once selected, we can look for sectoring. If this happens, we can take multiple Dikaryons in the same single cut. That's usually why sectoring happens, because we have numerous Dikaryons spreading apart from each other and creating their own space. um, so it will require further transfers to isolate into a pure or monoculture, right? So that's something totally uniformed, perfect growth around, no separation, no sectoring. Once our multi-spore isolate is producing uniform growth, it's time to get it into grains and cultivate. Now, if you are working with, I'm going to keep my space here. If you are working with a lab genetic that's already domesticated, once it's uniform, depending on how mutated or bottlenecked it already is, I would suggest taking at least another transfer before going to grains. If you're working with a domestication project, something that's more wild, you might want that early monoculture, early uniform growth to go in, because there may be some genetic diversity there that might express itself that you don't want to miss out on. So you don't want to over isolate, but you don't want to under isolate, because I'm sure everyone here has seen like a really sad basic multi-spore tub or bag, and the wide range of growth can be insane. I mean, you can have tiny fruits and massive fruits and all of it in the same space, and really sparsy flushing, and just there's too many reasons why you should isolate a little bit, right? I would suggest, if you're trying to look for genetic diversity and some different traits, start from spore and create a genetic 1, 2, 3, and 4, and then run all 4 simultaneously. So that way you can see if you're getting different traits from different Dikaryons or different sector growth. Um, and kind of looking for what you're looking for. So, all right, so you can isolate Dikaryons into monocultures if you continue this process, although it is quicker and more identifiable to grow and clone and get closer to a monoculture faster. So if you're in a hurry to just get to a monoculture, you can clone. It's a verified monoculture. Verified to a point. Fruits don't necessarily mean it is exclusively one Dikaryon, because fruits are made of mycelium. So if you're growing a lot of genetic diversity and you take a clone and that clone grows out on agar and it throws a bunch of sectoring and weird mycelial morphologies, it's because that fruit actually was made of numerous Dikaryons. So that's just something to throw out there as a little tidbit of information. All right, isolation is important for a few really good reasons. Consistency of flush, vigor, and speed. Vigor and speed mean you're going to outperform competing fungi or bacterias or something that can interfere with your cultivation. So you want vigor and speed to be part of it no matter what you're using or no matter what you're raising, growing, any of it. Because if you don't have vigor and speed, you have a weak Dikaryon and you're going to have to baby it. So you're going to have to be a little more adept in the lab, a little more careful, a little cleaner, because if it's slow, it's going to mean that it's going to be prey. So to aspergills or penicillins or something like trichoderma or some sort of mold. So you have to work really clean with slow genetics. I will definitely touch on that. We're getting there. We're so close. You guys are always right one step ahead. It's awesome. It is also important to remember the cellular structures of mycelium as it will guide you through much of the process. So these are just some basic visual aids for anyone who's like not stared at a million plates. They can grow in these weird shapes. So a lot of times when you get raised and convex or super umbonate or crater formed, a lot of that, if you're growing on agar, that is usually just too much nutrition. It's a great way to dial it back. But you should be flat and you should be in any of these other categories, right? Irregular, umbonate. Um, Irregular umbonate is usually like my ape. Filamentous is like half the library. Rhizoids like a few handfuls that just love to spider web out. Even if there's like, I don't know, just a few weird that it doesn't matter what the nutrition is. They just spider web out like crazy. So oh, absolutely. Have a good one. Thanks for coming. I will upload it and let you guys know. Over isolating play out. Yeah, so too many transfers before going to grain. Oh, it's the problem with that is that if you over isolate before you go to grain, you aren't going to be able to expand as far. So it just depends on how far that's supposed to go, right? So if I specifically because of what I do for a living, I can't over isolate. I'm always trying to find this like sweet spot because if I over isolate, then my culture is no good for someone who's going to expand it 10,000 times over, right? But if I under isolate, then it's not going to perform. So there is a sweet spot, and it's right somewhere in like this early stage. You just have to have clean work. You can really go to like a T10, T15, no problem. I've had great results going that far. So it's just something to kind of throw out there. Now, we're going to talk about like cutting plates, taking transfers. We saw some of those early cuts of spores with these sectors, right? So we have this Cabo as a great example, these nice triangular sectors. If it's me, I'm going to teach you the process in which I taught myself just from my own work. And this is something I observed of my work later. I didn't know I was actually doing this, which is really actually cooler to me as I like progressed, as I got further, my understanding of everything that I was doing, I kind of came to this like aha moment where I was like, oh my God, I didn't realize I was even doing that. So usually off of these sectors, because of their triangular shape, I take triangles for my T1, sometimes even a T2. And what it does is we can see here we're getting some sectoring, right? But this is probably contaminated. There's sectors here, but this is so weird and fluffy and irregular. Like I wouldn't trust this culture. I'm going to be trying to take some little quick, rapid transfers and see if I can clean it up. This one, though, is nice, clean, little sectoring going on. It doesn't like the triangle at the tips, but it's giving me these awesome canvases to take cuts, right? So let's say we're doing a T1 to T2, but I got this beautiful of a clean plate. I'm going to add a dimension to my cut, and I'm going to add in a square, which is here for whatever reason. Okay, so I got my square. We're going to add in this extra dimension, and it's going to create a little more uniformity to the growth, and giving it a little wider stance, smaller cut too. So, and typically the reason why I go from triangles here, triangle on this guy, in the beginning is because those sectors that come off these spore growths are in kind of a triangular shape. So it's a really great way to grab a large pizza pie of that genetic. And the reason why I'm grabbing a really large piece versus something that's small is because it's going to create a lot less stress on the genetic itself. And the reason why it's creating less stress is because we already know what's going on in our mycelium, right? We learned about what's happening inside of the mycelium, all of the cells, the organelles, the cytoplasm. Those cells work together. And if there's more of them, they can work together faster and better and healthier. It doesn't mean you're going to colonize a plate 2 days quicker. It just doesn't. But you might. Healthier? Absolutely. This is stressing, right? If you have a really great genetic that performs like a commercial grade genetic, it just can't slow it down, can't kill it. It's a stellar genetic. It just crushes everything. You can do these like tiny cuts and get these beautiful plates. Is it necessary? No. It's just not necessary. I think it's added unnecessary stress to the expansion, right? So we're going to take this tiny little bit of cells and force it to expand 1,000 times over for no reason. So if I'm always playing the expanding game. Like I have 1,000 cell divisions before I hit issues, right? Before I have sloppy flushes, before I have something not go right. Maybe it gets weakened because it's been alive too long, it's expanded too much, and it's going to trike out. So we're going to have those issues, right? So there is some things that you can do to eliminate that, and some of that is by just keeping more cellular availability. So when you're taking cuts, take big cuts, um, and just go with that. Okay. So how can you have contaminated growth without visible contamination colonies? Well, there are things called piggyback contaminations, right? So piggyback contaminations are things like trichoderma. Um, Trichoderma is a basically a parasite with specifically cubensis, so it will piggyback onto it. And you can look this up. There are types of aspergillus, and they will create coils around mycelium on a microscopic level. I really should put a photo of that in this presentation so you guys can see it. But you can Google it. If you Google aspergillus and you just search like coiled over mycelium, or if you do something like that in Google, you will find it. Okay. The trichoderma or aspergillus will create these coils around the mycelium, and you have a contaminated culture. It's literally weakening your culture as you grow it, and then once you throw it into grain, you're just gonna have a mess. So those are just things to be aware of. But if I am like super heavily isolating, trying to get to cell divisions quick, and I want to get expanded fast without going to T10, but I want to get there by T4, I'm going triangle, square, square, core, 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 right? Because this is going to force fast expansion, and I'm going to get a really super clean plate. Boom, straight in, right? You can also do trench tech. I put that out there like years ago. Trenching, you can outrun your piggyback contaminations. It's definitely the way to go. Trenching is great for that. What it'll do is the hyphae will grow across a plastic trench, take the agar out. The hyphae will stretch across it, and the piggyback can be like fuzzy on the back of it, kind of like slowing down its growth. You get a couple nice pieces of like clean tips of hyphae and transfer them really quickly, and you're good to go. All right, so next we're just going to go ahead and pop into a quick visual aid of just taking some transfers. Nothing fancy, again, just a visual aid for anyone who needs it. Dude, if you need a core, let me know. I have like 30 of them. They are not easy to. I ordered a bunch and they were on back order, and I got like 6 boxes one day. It was like, oh my god. So now I have like too many. So if you want one, just let me know. I always like to point this out. This transfer was like a really heavily thick poured plate. Sometimes you just gotta like, slice that sucker in half. Lose that extra weight. You don't need that agar because you don't want the mycelium pressed up against the lid. And paper cutters are great for wrap. Yeah, throw it in your notes, man. If you throw in an order or something, I'll throw one in there. Well, I don't do it for aesthetic on the mic side up. So the mic side up is actually for a lot of the stuff we were talking about, right? Like we've talked about mycelium functionality, why it grows the way it grows. And I will, here, let me get my chat back open. Okay. The reason why I put mycelium side up and not down is these photos are really great examples, visual, right? So I take this big cut and all of those organelles and mycelium within this connected network realize there's no more food around it. They start growing again and they form their new spikes and corpors and they start crawling down this edge. And the edge of that cut was not consumable before, so they're like food. And they send these pulsating signals throughout this whole cut. There's food all around us. And then they crawl down. And then they tap down their feet and they're like, oh my God, there's food here too. So now it grows very uniformly. A lot of times when growing with mycelium side down, you're not allowing this cellular process. So what ends up happening is sometimes you get these weird shoots. Even though you have a monoculture, and it's not growing uniformly. And that's because of the way it was put down on the plate. So that is the reason why I put mycelium side up.